So yeah, thanks again for organizing this conference, and thank you for inviting me. It will be a pleasure to give a talk. I will be talking about whole genome sequencing analysis, challenges, and opportunities. So first, I will give a brief introduction. Probably I'll do it very fast because the audience is familiar with this type of data. But I'll talk about what exactly is whole genome sequencing, how is it different to other techniques, sequencing and genotyping, and what are the costs associated with whole genome sequencing. And then I'll talk what kind of value provides whole genome sequencing and what are the challenges associated with this type of technique and analysis. And yeah, in particular, I will talk about imputation, population structure in rare variants, and rare variants grouping and analysis methods in general. So, you probably know the central dogma of molecular biology and what place DNA has in it. And during the last 10 or even more years, genome-wide association studies have been widely successful because our genome is fixed from the birth. I mean, of course, there are also other mutations such as somatic, which are cell-specific, but for this talk, I'll focus on germline mutations. And currently, there is also a lot of other omics data which is being produced besides genomics, transcriptomics, proteomics, as you heard from the previous talk. But those type of data, first of all, they are tissue and cell specific, and second, then they can differ longitudinally, so among different time points. And genetic data is fixed, and this allows us, this is the reason why it's extremely popular to work with genetic data. So, and our human genome consists of 3.2 billion base pairs and contains more than 20,000 protein coding genes. So you can imagine how this is a huge part of the big data, which is pretty popular right now. But now just let's take a step back and see what other methodologies there are. So the most popular technique to do association studies, even now, is still array genotyping. And depending on the array size, you basically read specific positions from your human genome, and this can range from 500,000 up to 2 million base pair positions. And usually those positions cover some common mutations, common variation. And other missing variants from the human genome can be imputed based on some imputation panel. And this is possible due to the correlation structure which exists in our human genome. It's called linkage disequilibrium. And some large consortia of complex diseases have developed even specific chips. For example, there is a schizophrenia chip, which also includes some rare variant which have a value for specific consortia. And basically the largest and latest data sets include up to 500,000 or even more individuals for which there is genotyping data available, for example, the UK Biobank. Yes, and recently, this also includes UK Biobank, consortia started producing whole exome sequencing. And this is popular because basically this is a trade-off between the cost of sequencing the whole genome and covering the low-hanging fruits, the functional exonic variants, which are the most important for biological researchers. And basically it covers 1% of the human genome, but it contains on average like 1-2 million mutations. Most of them are pretty rare. Now coming to whole genome sequencing, it reads almost every base pair of the human genome. So it has the advantage to study all the mutations, including those which are located in non-coding regions. And it also has some advantage for the exomes, because it has some, as it was shown, it has some better coverage on the edges of the exome, where you can also find some functional mutations. As I mentioned, for whole exome, this is also true for whole genome, most of the discovered mutations, discovered variants, are rare. When people say rare, they usually mean population minor frequency less than 1%. But if you look at the distribution, it's even, so you see that the vast majority of the variants are actually present in the data set once. And in the table, you can see how the number of variants differs with the sample sizes. This is taken from the currently largest whole genome sequencing initiative, TopMed. So you can see how quickly this types of data set grow. And coming to the cost of whole genome, this is still pretty expensive, but if you look at the last 20 years, starting from the first sequenced human genome, which costed hundreds of thousands of millions dollars, and now you can sequence your genome for less than $1,000. It's actually a pretty impressive way which was being made. So although genetic data is pretty simple, it's basically a matrix consisting of zeros, ones, and twos when you call your variants. There is no agreed format in the community of such data. So there are multiple widely used formats, and so they have their own advantages and disadvantages. And also historically, people from different research groups stick with one format, while others stick with the other. So on the upper left side, you see basically the sequence alignment format, and then the other three formats are already variant calling formats: the VCF, BCF groups, the PED-BED coming from the Plink format, and the GenBgen, which is coming from the Oxford-based tools. Okay. Yeah, so let's talk a little bit about heritability. And so what is actually heritability? Heritability is the phenotypic variance which is attributable to genetic factors, and there are multiple definitions for heritability. So the most broad, it's called the broad sense heritability, is what I said, so variance attributed to genetic factors. But there's also the narrow sense heritability, which is basically the phenotypic variance attributable to additive genetic factors. This is the lowercase h2. And then, so the most widely used is actually the observed heritability, or the SNP heritability. And this is the phenotypic variance attributable to observed SNP, observed variants. And yeah, so as you can see from the plot, this is an example for different complex diseases, how much estimated heritability from how much heritability can be explained by genetics from twin studies, and how much was explained by GWAS. So in the dark red color, you see heritability explained by genome-wide significant variants. In the red, or I don't know, salmon, in the red color, you see the total heritability, which is captured by GWAS, but still not significant, so to say. And then by the salmon or light red color, you have the missing heritability, which is yet to be found. And there, basically, whole genome sequencing can help us. So, okay, so where does the missing heritability come from? So it can be attributed to common variants of low effect size, and with the growth of the recent sample sizes, those variants can become genome-wide significant, and those are the ones in the red color in the previous slide. Then there are the rare and low frequency variants, as I already mentioned. They are usually identified with either exome or whole genome sequencing, and except very large effect sizes, such as for example, the ApoE variants in Alzheimer's disease, the E2, there is a protective allele. You have a similar problem as in common variants. You need to increase the power to identify such variants. And other possible reasons include some structural variations, gene-gene interactions, non-additive effect, and maybe the heritability from twin studies was simply overestimated, for example, by shared environment. So how whole genome sequencing can help to fill the missing heritability? So I will focus on two things. So first, whole genome sequencing can definitely help to build larger and more diverse imputation panels for GWAS studies. And I will also try to answer the question whether imputation could actually replace the need for whole genome sequencing. And second, like I was saying, a very important part in whole genome sequencing studies is rare variant analysis. And I will talk about this because whole genome sequencing can be important to study gene regulatory regions, CNV structural variants, and other non-coding variants of unknown significance. So I will focus on three challenges. The first one is related to imputation. So probably all of you know that imputation is a statistical inference of unobserved variance based on the correlation structure, the linkage disequilibrium. This is basically the horizontal structure in this plot. And in order to perform imputation, you need your genotype data set, which you want to impute, and the reference panel, which is based on whole genome sequencing against which you want to impute. And the question is how to choose this reference panel and how large it should be. Well, the most popular reference panels are now in this slide. So there is a very popular one is the 1,000 genomes with 2,500 individuals of diverse ancestry. You have an African specific panel, so with African ancestry, it's called Kappa, and it's not too large. Then you have the HRC, the Haplotype Reference Consortia, which is pretty popular. It has 32,000 individuals, and most of them are from European ancestry. And then most recently, TopMed has introduced their reference panel with almost 100,000 individuals and of diverse ancestry. Besides diverse ancestry, TopMed is not a population-based cohort. It actually contains multiple studies of different heart, lung, and blood diseases, and this might also help imputation, in particular if you study the same disease as those cohorts are based on. So there were some analyses which compared the reference cohorts, and for example, in the plot above, you see there were two studies of African ancestry, the GHS, and of Hispanic ancestry, the HCHS, and they were imputed against 3 different imputation panels. And these are the results, how well the rare variants are imputed, because basically those are the most problematic variants when you do imputation. So, and as you can see, size matters, but also diversity matters. You see that TopMed performs among the best, but also 1,000 genomes shows better performance since it has more diverse ancestry. Yeah. So could actually imputation replace the need for whole genome sequencing? I mean, if you have a large enough reference panel, why don't you just do genotyping and then impute against this panel and get all the variants you need? So to answer this question, I actually compared real sequenced whole genome sequence variants to the one which were inferred with imputation. And those were coming from the same cohort, from the same subjects. So, and as you can see from this box plot, actually still a large chunk of rare variants are specific to whole genome sequencing. So still, even with large cohorts, I think this was based, this was based on HRC matched ancestry. You cannot detect all the rare variations there. And also another thing, if you see in green and blue, that even for common variants, there is a small proportion of discordance between imputed and sequencing variants, which means basically that some of the imputed variants do not match with their actual real sequenced variants. Another interesting observation was that in long-range LD regions, so those are regions where you have a very long correlation structure, although those regions are actually pretty well imputed for common variants, but the quality, the imputation quality score for those variants shows to be pretty low. So maybe some of those variants are actually filtered out during quality control. And so the next challenge is population structure. And population stratification, this systematic bias, which affects large association studies. And this also affects rare variant association studies. So, and here you can see, so from this plot, so this plot is based on 1,000 genomes, and this plots the co-occurrence of variants which are present only twice in the whole cohort of rare variants. And as you can see, those variants are actually present within the same population or within the same supercontinental, so within the same continent. So basically, rare variants show more recent population structure, but regular methods which are based on the genetic variance covariance metric or the GRM, they are not able to capture rare variant population structure. So we have done some work to use alternative similarity matrices instead of the genetic variant covariance metrics to account for population structure. So basically, instead of the GRM, to construct principal component analysis, one could use the Jaccard similarity matrix, which is actually widely recognized in other fields. And if you do this only on rare variants and compare to the regular PCA on the GRM, you see that the Jaccard matrix actually performs better. And the reason is that it's the way it's made, it emphasizes mutual presences in sparse data, which rare variants are basically. It's just a large sparse matrix. And we have also successfully applied it to rare variant association studies. And more recently, this method was also applied to identify regional population stratification. And also it was applied to study COVID-19, and basically not to human genomes, but to viral genomes. So it was able to cluster the viral genomes based on the GSAID database and to cluster them according to the different clades. And so the third challenge is rare variant analysis itself. As I said, given the rareness of the variants, there is no statistical power to detect them unless the sample size is really huge. But what you can do is you can combine the signals from several rare variants and test basically the burden of this signal. And then you will get more power to identify the, depending on the effect sizes of those variants and the proportion of causal variants among those variants which you combine, you will achieve more power. And so there are a lot of testing strategies developed during the last 5-10 years. And so you can, all those testing strategies you can classify into 3 categories: burden tests, and as the name states, you test the burden of multiple variants. And there are different modifications based on some weighting schemes, so minor frequency stratification, and those tests are powerful for signals where you have consistent effect directions, so where all rare variants tested are pointing are either risk or protective, so have the same effect direction. Then there is a group of variance component tests. Most popular is SCAD, sequence kernel association test. There is also a version for combining rare and common variants of this test. And it jointly tests the variance component of the individual variants. And it is, so the main advantage is that it's robust to effect direction, but it comes to a cost that it's less powerful for regions with consistent effect directions. And then there is also a group of combined tests where those tests try to combine the advantages of the burden and the variance component test by computing the weighted average and exploring the space of weights for those 2 tests. Those tests are more robust, but they also come at a computational cost. So although rare variant testing is basically well established, grouping strategies are not so straightforward, and there is still research going on how to group the rare variants which you want to test. So for exome studies, besides sliding windows, people use functional annotations. So you can test all loss of function variants within an exome, or you can broaden this group and include stop loss, missense variants, splicing variants, insertions into this group, and test them together. This is perfect for whole exome sequencing. But how about whole genome sequencing? So for whole genome sequencing, people mostly use window-based approach, so either sliding window based on a fixed length or a fixed number of variants. Those sliding windows can be consecutive, they can also be overlapping. And you can also base them on promoter or enhancer regions. So I did a quick simulation when I was exploring the power of different grouping strategies, and here the line corresponds to the percentage of risk variance within a window where you test the whole window. And that is, the larger your region is, the more null or the more noise variance you include in a region. So as you can see, even though the power depends on the sample size, it is also important to include as much causative variance as possible. So basically, there's a trade-off between this window size and sample size. However, a priori, you don't have the information which of the variants are causative, else it would be pretty simple. So in terms for exome sequencing, you can make an educated guess for this by including some annotated variants which have a functional effect. Another thing what we have done, we tried to look for a data-driven approach to group the rare variants, and in this paper we have developed basically a method which looks at the spatial distribution of rare variants along the chromosome. And it groups densely located rare variants. So the window size is actually different for different groups. And the cut points are located in sparse regions where you have less variation. So, and we applied this this method to Alzheimer's disease and identified a region-based signal in the ITGB3 gene, which is associated with Alzheimer. And again, you can also use external annotation for whole genome sequencing. For example, you can make an educated guess based on GenoSkyline scores. So those are scores which are calculated on the Epigenomics Roadmap project, and they combine information from expression data, ChIP-seq data, DNase-1 hypersensitivity data. And basically, by studying some complex disease, you can identify the tissue of interest and weight your rare variants based on these scores. So, and yeah, here is also a bonus challenge which we faced recently. So this paper is basically about a gene-environment interaction analysis. So it's a sex-specific analysis of Alzheimer's disease using family-based design and whole genome sequencing data. And we tested the joint effect of Alzheimer association signal and sex interaction. And what we found, so in this Manhattan plot, you have the red signals, which are pretty significant, but those are actually false positive because, as we found, they were either mismapped from the X or Y chromosome within an existing pseudogene on this chromosome, or located in some repeat regions of retrotransposons. And I mean, we went into the quality control of whole genome sequencing data, which we have, and it was very good. It looked very good, but I guess based on the read size of when you do the whole genome sequencing, you still can face this particular problems, especially when you look at such an environmental effect as sex. Okay, so, and to summarize, so we talked about imputation panels. Whole genome sequencing should provide and is already providing more diverse imputation panels. However, imputation cannot fully replace the need for whole genome sequencing in rare variant association studies. Rare variants can help to explain heritability, but most likely they have moderate effect sizes. So as popular in genetics, larger sample sizes are required. Also, rare variants capture recent population structure, and I have shown that alternative similarity matrices, which are sensitive to sparse data, can capture the structure pretty well. And for window choice for rare variants, you need a more informative choice. And this, coupled with integration with other omic data, may enhance whole genome sequencing rare variant analysis. So this is basically it. I'm happy to take questions. Those are my acknowledgements. Thank you.